Hi guys, this is a review video on the basal ganglia. This is more neuro review. Specifically, this is also coinciding with lab 7 on the basal ganglia. So just to reiterate, because I think it's important to be said, this is really hard stuff. So, um, you we're doing our best, and, I hope you guys are doing well too. All right, that said, we are a tiny bit tired. It's fairly late, and we're just trying to cement this information for us before we get too tired. So, all right, starting off with the basal ganglia, what is the actual function here? The idea is that the basal ganglia is going to take in basically data from or action potentials from the cerebral cortex, and it's going to amplify motor movement or decrease motor movement, right? And so it's going to influence the motor behavior of different motor pathways, and these are ones that we've started to become familiar with. We have the cortical bulbar and the cortical spinal. These are the main two that the basal ganglia are going to act on. Why? Because you recall that these two are in fact voluntary muscle movement tracks that we talked about in the previous lab 6, right? And then reticulospinal, reticulospinal, and tectal spinal are going to be dealing with balance and head and neck, in addition to cortical bulbar, which is also head and neck. All right, so amplifying and increasing motor movement via something called the direct pathway, which we'll get to in a minute. Decreasing or sort of stopping motor movement via the indirect pathway. And then we have this third pathway, which is only a single step, which we love, which is the nigrostriatal pathway. And so this is going to modulate the direct pathway, which is going to direct amplify movement. And then it will decrease the indirect pathway, and our indirect pathway is our brakes pathway, okay? And it's going to do this by producing dopamine. So dopamine is going to be our modulator, and we're going to see its effects in this nigrostriatal pathway, okay? But basically, generally, our basal ganglia is doing sort of a tonic baseline inhibition of excessive movement, but we can sort of stop that inhibition via the use of dopamine. All right, so we have the case of this particular lecture. I think was very well taught by Dr. Walker, and we have this nice diagram that we'll get to in just a minute. I've broken it down a little bit differently than how he did. He just gave us this whole diagram all at once. I broke it down such that we build up to the diagram that he gives us. So this one's the one that Dr. Walker gives us, and then these two are the individual pathways that lead to this guy, which I think, at least for me, was an easier way to think about what was going on. So what exactly is the basal ganglia, or where is it located? So it's located, here's our friend the cortex, here's our corona radiata, and here is our friend the basal ganglia, right? So it's deep to the corona radiata, in the cortex, and it's actually made up of 4 main components. We have the striatum, and the striatum is Latin for striped. It's made up of 2 nuclei, the caudate nucleus, which is Latin for tail-like, and then the putamen, which is Latin for husk or shell. And so that's located here. This is the caudate next to the lateral ventricle. And then this is the putamen right here. All right, so together, these two make up our striped, or our striatum, which we will abbreviate as STR. Okay, STR for striatum. All right, so that's this is a nice sort of cross section. This is my favorite diagram view of the basal ganglia. I think it's nice and easy, but it actually looks a little bit something like this. So we said that putamen is like husk or shell, right? So that's this region right here. Then we have caudate is tail-like, right? So attached to the putamen, we have the caudate nucleus, which is tail-like, right? Doesn't it kind of look like some weird alien? And then deep to the putamen, the husk or the shell of the basal ganglia, we have the space back here, which is cleft for the internal capsule, which we're familiar with because it has the posterior limb and the anterior limb in the genu, which you may remember from the cortical spinal and cortical bulbar. And then we're going to get to more of the other nuclei, but we said again there's 4 main components of the basal ganglia. The first is the striatum, which are made up of 2 nuclei, which are functionally the same. We have caudate nucleus and putamen. At least we're treating them as functionally the same in this class. All right, our next main component is the globus pallidus, which is Latin for pale globe, and that is located right here. In fact, it has 2 nuclei as well, which are functionally distinct. The first is the globus pallidus externus, which we will abbreviate as GPE, and then the globus pallidus internus, which we'll abbreviate as GPI. Okay, and so going back to our nice diagram of the basal ganglia, we have our putamen, the husk or shell, we have our caudate nucleus. Together they make up the striatum, and then on the sort of medial to this, on the other side of this husk, and it's outlined in dots because it's on the other side of this. It's not like a bump off of this laterally, but medially, right, deep, we have the globus pallidus, right? And so that's easy to see here. Here we have the putamen, the husk or the shell, right? And then sort of wraps around, and you get to this guy, right? We're seeing a cross section here, and then we have global pallidus externus, global pallidus internus, all right? And they have two distinct functions. All right, so this is the first component, second component, and the first component has two nuclei. The second component also has two nuclei. All right, then we get to the subthalamus. This is our third main component of the basal ganglia. And here we have the thalamus, and so the subthalamus is quite literally that sub below the thalamus. So the subthalamic nuclei. This will be part of our indirect pathway, which we'll get to in a minute. And then here's where the magic happens. We have the substantia nigra, which is Latin for black substance, right? Substantia nigra, black substance. All right, and so you're like, why would it be called that? Well, here's a cross section of the midbrain, and this part right here is the substantia nigra. And notice how it's dark. That's why it's called the substantia nigra. But it actually itself contains two functionally distinct nuclei. We have the pars compacta, and this guy's going to be our nice workhorse for modulating movement, as we'll see, which we will abbreviate as SNC, C for compacta, and then SN for substantia nigra. All right, and that's located right here. This is in the midbrain. All right, so substantia pars compacta on the top, and this is going to be the one that produces a whole bunch of dopamine. All right, so this is our dopamine section. It's the part of the substantia nigra. This part right here, the darker part is in fact the pars compacta, and then adjacent to that we have the pars reticula. Okay, but it's not as dark. It should be white. All right, but pars reticula is the second part of the substantia nigra. All right, but the basal ganglia overall, we have the striatum, which we'll abbreviate as STR. We have the global pallidus, which has two parts, GPE and GPI, functionally distinct. Then we have the subthalamus, which we'll abbreviate as STN. And then we have the substantia nigra with the pars compacta, which is the dark region. And then we have the pars reticula, which we'll abbreviate as SNR, and pars compacta as SNC. All right, so just a tiny bit more. We think to ourselves, well, if dopamine is what our pars compacta is going to be dealing with or producing, and so like why exactly is this substantia nigra dark? Well, here we have tyrosine. You can follow this down, this pathway, biochemical pathway down to dopamine, but you can also take it and produce melanin. And we know melanin being the pigment in the skin that is brownish, right? And so basically this dark substance is our melanin pigment from the fact that the pars compacta does a lot of dopamine production, and a lot of this precursor pathway you can also go to melanin. Do you need to know this? No, it's just, why is it dark? All right, so now we're starting to get to the actual meat of the content. So again, we said we are amplifying motor movement via something called the direct pathway, and then we're going to reduce motor movement via something called the indirect pathway, and then we have this nigrostriatal pathway, which is going to add dopamine, which is going to change how much of the direct pathway is going to be expressed, and how much of the direct pathway it's going to stimulate the direct pathway and decrease the indirect pathway. All right, so what's this direct pathway? What's this indirect pathway? What's this nigrostriatal pathway? It's really not too bad. Starting with the direct pathway, I've put it in red specifically because it's stimulatory, right? And so the goal of the direct pathway is to increase or amplify movement. All right, and so the steps are relatively straightforward. We have from the cerebral cortex, right? Remember, we have the motor cortex on the cerebral cortex, where you get your voluntary muscle movement. So we have afferent incoming stimulatory signal coming into the striatum, STR. Striatum, remember, is the caudate nucleus and the putamen, and those are located, again, right here and right here. We treat them as functionally the same for the purposes of this class. And then, so we have incoming afferent signal. It's going to use glutamate at the end of its axons, that synapse on the striatum. After stimulating the striatum nuclei, now stimulated, will release more of their neurotransmitter, which is GABA, and GABA is an inhibitory neurotransmitter. And so GABA is going to act on SNR. We said SNR was substantia nigra reticulata. Right? It's right here, substantia nigra reticulata, pars reticulata, and that is located right here, part of the substantia nigra, and the global pallidus internus. All right, and so you have your inputs, cerebral cortex, you have your two basal ganglia nuclei in the direct pathway, and then you have your substantia nigra reticularis and the global pallidus internus are going to send axons out to the thalamus, specifically the ventral anterior and ventral lateral nuclei, to the peduncular pontine nucleus, PPN, and then the superior colliculus. All right, and specifically it's going to send out axons that will release GABA. And then, so each of these targets themselves, the thalamus is going to send out axons up to the cortex, where it will synapse with the pyramidal cells that are dealing with the voluntary movements of the rest of the limbs, the appendicular part of the body, and that's the cortical spinal tract responsible. So the pyramidal cells, remember, give us the cortical spinal tract, and then the head and the neck muscles, the cortical bulbar tract. All right, PPN, peduncular pontine nucleus, is going to send out axons which will release acetylcholine, stimulatory neurotransmitter. It's going to act on the reticular spinal tract to do postural movements, axial movements, and then superior colliculus, you remember from the octave, like when we talked about the eye. So it's going to release glutamate, which is stimulatory neurotransmitter, and basically act on the eye movement centers, and then the tectospinal tract as well for head and neck muscles, in addition to the head and neck muscles that are innervated by or controlled by the cortical bulbar tract. Okay, that was as clear as mud. I'm so sorry. Let's just try to big picture it real quickly. So direct pathway is to increase, amplify movement. Its inputs are the cerebral cortex that will stimulate the striatum nuclei of the basal ganglia, which is the caudate and the putamen. This being stimulated is going to, it uses GABA. It's going to send out more GABA to the substantia nigra pars reticulosa and the global pallidus internus. And so if this guy's been stimulated, it's going to release more GABA. GABA is inhibitory, so this guy's going to be more inhibited. Right, that's what these sentences down here are saying. And then if this guy's more inhibited, it's going to release less GABA overall. Less GABA on each of these targets means that they are less inhibited, and therefore you're going to have more of the glutamate, more of the acetylcholine, more of the glutamate released. And so all of these guys are going to have more movement. All right, so the direct pathway, which is actually only this part right here, from striatum to pars reticulosa and global pallidus internus, has a net result of more movement. Okay, so let me just read the steps again. The afferent stimulatory input from the striatum, aka the caudate, afferent stimulatory input to the striatum, aka the caudate and putamen, it's getting that from the cerebral cortex, and it's going to have glutamate act on this striatum. The striatum nuclei, now stimulated by the glutamate, will release more GABA on inhibitory, it will release more GABA onto the reticulata and the globus pallidus internus. More GABA on these guys will mean they will be more inhibited, and they're going to release less GABA themselves, and that will mean that their targets will therefore be quote-unquote stimulated because they're less inhibited. All right, if these guys are stimulated, then they're going to release more of their stimulatory neurotransmitters, and you're going to get more movement overall. All right, so the direct pathway is giving us more movement. The indirect pathway is the opposite. Right, we said that this part right here is technically the direct pathway, just this particular step from striatum to globus pallidus internus. The indirect pathway is this part right here. All right, so notice we're going from striatum to pars reticulosa and the globus pallidus internus, just like in the direct pathway, but now we have additional steps. All right, and so the goal of the indirect pathway is to decrease movement overall, all right, and to prevent excessive movement. So just like before, you have input, you have afferent stimulatory input to the striatum. aka the caudate and the putamen of the basal ganglia, and it's coming from the cerebral cortex via the stimulatory glutamate that the cerebral cortex is releasing. So the striatum has now received this stimulatory glutamate. The stimulated is now going to release more of its product, GABA, and it's going to release more inhibitory GABA on this nucleus right here, which is the global pallidus externus, which we abbreviate as GPE. All right, again, that's right here, global pallidus, this one, GPE. All right, so more of this guy means this guy is more inhibited. So more inhibited global pallidus externus releases less inhibitory GABA, right? It releases GABA onto the subthalamic nucleus. And so less inhibited subthalamic nucleus means that it's going to release more stimulatory glutamate. So this guy releases glutamate onto this guy, right? And so if you're releasing more stimulatory glutamate onto the pars reticulosa and global pallidus internus, that means that they are going to release more GABA. We know GABA is inhibitory, and so you're going to have inhibition of each of these guys, and so they're going to release less of their products, and so the net result is you're going to have less movement overall. All right, so the net result of indirect pathway is less movement. All right, so big picture, we have direct pathway, more movement. It's literally just this particular step. Indirect pathway is this step right here, and indirect pathway is going to do less movement overall. All right, but still, we haven't added our dopamine yet because the dopamine is going to do modulating. All right, so we have this nigrostriatal pathway where the goal is to modulate the stimulation of the direct and indirect pathways, right? Direct, increase movement, indirect, decrease movement, act as a brake. So the effect of dopamine, we're going to release dopamine onto the neurons that are in the first step of the direct and indirect pathway. And what is that? Well, it's the striatum, right? This is the first step of the direct and indirect pathway. So basically, what is going to produce the dopamine? Well, it's our substantia nigra, specifically the pars compacta of the substantia nigra, right? We know it's the dopamine center because we said that dopamine and melanin production are closely related, and this part of the midbrain is very dark, right? Because there's a lot of melanin because we have the production of a lot of dopamine. All right, and so the pars compacta is going to release dopamine onto the first step of the direct pathway and indirect pathway. All right, and so the idea is that dopamine on the direct pathway, well, the direct pathway neurons in the striatum, they're going to express a D1 receptor for dopamine, and the D1 receptor is actually a stimulatory receptor. And so when dopamine acts on the direct pathway, it's actually going to stimulate the direct pathway. Well, we already know what the effect of this direct pathway is, right? That's more movement. So if you're stimulating the direct pathway, you're going to just increase the amount of movement. So dopamine will increase the amount of movement by increasing the direct pathway. Its effect on the indirect pathway is different. The indirect pathway, the cells in the striatum, again, what is the striatum? The putamen and the caudate nucleus of the basal ganglia. The indirect pathway, the dopamine, is going to activate D2 receptors instead in the indirect pathway at the striatum, which we abbreviate as STR. All right, the result is you're going to have a decrease in the direct pathway because D2 receptors are inhibitory. And if you have less indirect pathway, that means less braking, and you're going to have more movement. All right, so we'll get to the diagram in just a second, but our takeaway for the effect of dopamine for the nigrostriatal pathway, which is literally just the step from the pars compacta to the striatum. Why is it called nigrostriatal? Well, this is the target, right? Striatum, and nigro for substantia nigra, right? Which is the pars compacta is one of two parts of the substantia nigra. Okay, so the takeaway is more dopamine means stimulate the direct pathway, so you're going to get more movement, and you're going to inhibit the indirect pathway, and so that will mean less braking. And so the net result is more movement, right? If you have less dopamine, then you're going to have less stimulation of the direct pathway, and you're going to have less inhibition of braking, so you're going to have more braking, right? So you'll have less movement overall with less dopamine. All right, so let's just take a look real quickly at our overall diagram. So we have our inputs to our basal ganglia. The overall input is our cerebral cortex is like, hey, I want to make a move. I want to move my body in some shape or form. And it's going to send out some glutamate, and it's going to stimulate the direct pathway, and it's going to stimulate the indirect pathway at the same time. All right, and so they kind of cancel each other out because we know direct pathway is going to cause more movement, indirect pathway is going to cause less movement. All right, but what if we were to add our third pathway, the nigrostriatal pathway, right? And so this is why the pars compacta is really important, because our inputs are saying, oh, well, this is what we want to do, but the direct pathway is increasing, indirect pathway is decreasing, so that cancels out. But what if I were to increase the, like super stimulate the direct pathway, and and then decrease the indirect pathway? That's what our nigrostriatal pathway is doing. So we have our dopamine being released. For the direct pathway, it's acting on D1, so it's stimulating, and then on the indirect pathway, it's inhibiting the indirect pathway overall. All right, but again, this right here is the direct pathway. This right here is the indirect pathway. All right, and so all of this information, you really just have to write it out a few times, like copy this chart. And so in this review, this is the thing that Dr. Walker gave us. And so just, filling this out a couple of times, we have input from our cortex. We know that's going to go to the striatum. Then for the direct pathway, we go to the global palatus internus, and we go to the substantia nigra reticularis. Then we're going to go to the thalamus. Then we're going to go to the PPN, and then superior colliculus. And we know that this is going to be voluntary movement of the head, neck, and like appendages. And this is the pyramidal tract. And then this one is going to be reticulospinal tract. And it's going to be about balance. And then this one's going to be the sort of ocular sort of vision, like eye movement. And then also head and neck movement with tectospinal. Okay, and then we know this is the subthalamic nucleus, and this is global palatus externus, and then this is our friend substantia nigra pars compacta. All right, and then we know that the cortex is saying, hey, I'm doing something, I'm going to stimulate you, so this is glutamate. Then we have GABA. Then we have GABA, 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 all inhibitory. Then we have more GABA, and you guessed it, more GABA. And then right here we have a glutamate. And then here we have glutamate. Here's ACH. Here's glutamate. And then here we know is our friend dopamine. And we know DOPA acts positively on D1/direct and negatively on D2/indirect. All right, so practice it a couple of times, and have it down. Okay, but this is sort of the net diagram that Dr. Walker gave us. Once you know what it is, it's pretty useful. Direct pathway, again, expresses this D1 stimulatory receptors, and it promotes movement, right? That's this pathway right here. Indirect is this guy, and it acts as a brake on these guys overall because we have this GABA, which is inhibiting these guys. Okay, and our brake is stimulating the production of GABA. All right, so then we need to know basically all of these cross sections. This is in our lab, so I would say, try to go through it quickly, but again, what are the parts of the basal ganglia? We have our striatum right here. Then we have our global palatus externus, internus. We have our subthalamic nucleus. We have pars compacta, pars reticulosa, and those are part of the brainstem. And so what we'll be given is like slices through the brain like this, and we need to be able to determine the different parts of the basal ganglia, because we want to be able to, one, draw this out, but then two, say, okay, well, what happens if I get rid of dopamine, right? If I get rid of substantia nigra compacta and I get rid of my dopamine, you're like, oh, well, we know what dopamine does, right? So we have our more dopamine means more movement, less dopamine means less braking, or more braking, less inhibition of the indirect pathway, all right? But the idea is we draw this out, we look at these guys, find our lesions, we know which part of this diagram has been taken out, right? All right, so then first off, we have cross-section here. We're pretty, towards the front of the brain, and we have the head of the caudate nucleus right here. This is the anterior limb of the internal capsule. This is the putamen of the basal ganglia. This is the anterior horn of the lateral ventricle. All right, remember the putamen and the caudate are connected to each other. Putamen is husk, right? Putamen, and it wraps around and you get caudate. Okay, so moving on, then we're a little bit further back now, and we have putamen. We have the first appearance of our global palatus externus. This is the posterior limb of the internal capsule now, no longer the anterior limb. Here's the body of the caudate, body of the lateral ventricle. Here we have corpus callosum, and here we have the thalamus. All right, and then here's our interior commissure. All right, then moving on, we have putamen. Again, we're moving more this way each time. Putamen. We have global palatus externus, internus. This is posterior limb of internal capsule. Here we have thalamus. In particular, we have the ventral anterior thalamic nucleus. And you may recall that the thalamus targets from the pars reticulosa and global palatus internus are ventral anterior, ventral lateral nuclei, right? And so here we have, once this thing loads, we have ventral anterior thalamic nuclei. All right, so moving on, again, go more this way. Putamen, global palatus externus, internus. Here we have ansa lenticularis. This is one of the one of the axon branches. Then we have posterior limb of the internal capsule, and then this is ventral anterior nucleus of the thalamus again. Putamen, external, global palatus externus, internus, posterior limb of internal capsule. Here we have substantia nigra, subthalamic nucleus. This is the thalamus, so that makes sense. And then lenticular fasciculatus, which I didn't specify before, but I actually have it marked here. That is the axons between the global palatus internus and the thalamus. Okay. All right, so then here we have midbrain, cerebral peduncles, substantia nigra. Okay. And then here we have the pedunculopontine nucleus, PPN, which is this guy right here. And then finally, we're almost done. We have putamen right here. Then we have thalamus right here, caudate. And then we have, so putamen right here, and then global palatus. And so one of the things they mentioned in lecture was this kind of looks like a lens. Theoretically, maybe it does a little bit. And so we call it the lentiform nucleus or the lenticular nucleus, the putamen and the global palatus together. Again, that's this right here is supposed to look like a lens, so the lentiform nucleus. All right, so we're almost, I think we're just about done. We need to go over Parkinson's, but we're just about done. All right, so here we have midbrain, and again, we're looking at the substantia nigra. We know that it is that because it's nice and dark, and the dark spot in particular is the pars compacta of the substantia nigra. All right, but we like it because we know substantia nigra, and you're like, something went wrong. We have less dopamine. Well, we know more dopamine means more movement, less dopamine means less movement. So that's what we're going to see with Parkinson's. But let's start talking about our disorders. So before we could do that, though, we have the nice circle of Willis to talk about, blood supply to this region. We have the basilar artery coming up, and then substantia nigra being supplied by that, posterior communicating, posterior choroidal, subthalamic nucleus, substantia nigra, posterior communicating, subthalamic nucleus, and yeah, and then basically this is our circle of Willis, and you could draw like the other part of it, right? We're seeing half of it, but we have basically just the central carotid coming up, and then we're getting to basically all of this is meant to be sort of, if you have a blockage, then we could have potentially, as we'll talk about hemibolismus, if we have a blockage here and here, then we're going to kill off the subthalamic nucleus. Subthalamic nucleus dies, then we're not going to have basically all of the indirect pathways gone. If all of the indirect pathway is gone, then there's nothing stopping excessive movement. And so hemiballismus in particular is occlusion of the subthalamic branches arising from the posterior communicating or posterior choroidal branch, and you're going to have excessive movement. So hemiballismus is like one-sided ballistic movement, ballistic being like excessive movement, right? And it's because the STN has been knocked out. There's no more braking, no indirect pathway. All right, Parkinson's is, I imagine, what we're going to get a question on an exam, at least I hope so. You have this mnemonic trap for the symptoms, and that's tremor, rigidity, akinesia, so less movement, postural disturbance. And so the idea is that you have less dopamine. For whatever reason, your neurons in your substantia nigra, specifically pars compacta, have kind of decreased. And so this is something that's supposed to happen with age generally, but in Parkinson's disease, you have more of it. All right, and so in lab, we were given a nice cross-section like this, and we were told, well, on one side, it looks like we have less substantia nigra than on the other side. And so on this side, you're going to have you're going to have contralateral to this side, you're going to have decreased movement overall. All right, so for all of these, it's supposed to be contralateral is where the symptoms show up. All right, but if you have less of this, you have less dopamine. And we know what happens when you have less dopamine. Well, less dopamine means more braking, right? More dopamine means more movement because it stimulates the direct pathway and inhibits the indirect pathway, inhibits our brake. So less dopamine means less inhibition of our brake, which means less movement, right? And that's in fact what we see with our Parkinson's disease, all right? But there's something wrong with this substantia nigra. All right, we already talked about this guy. Huntington's, basically we have an issue going on here, and what is in that particular spot? Well, let's take a look once it loads. All right, we know that here, adjacent to the lateral ventricle, is the caudate, right? And so caudate nucleus is, well, that's part of the striatum, right? So in Huntington's disease, you have this guy knocked out. And so basically you have, if that guy's knocked out, then you have hyperkinetic movements of Huntington's disease, and it's supposed to be a little bit unclear as to why, if you have this whole thing knocked out, then the direct and indirect pathway are affected. All right, and then finally we have Wilson's disease. This is called hepatolenticular degeneration, autosomal recessive affecting ATP7B, okay, gene involved in copper uptake. And so the idea is that this liver gets damaged, but in particular the lenticular nucleus, which we know is the thing that looks like a lens, aka the putamen and the globus pallidus, is damaged, and so is the thalamus. And so putamen damaged, that's striatum overall. So you have issues with direct and indirect pathway, and so basically you're going to have like abnormal movements, as you can see here. It's kind of similar to Huntington's. You have hyperkinetic. Wilson's, you have damage to lenticular, but you have these abnormal movements overall. All right, so here again is our practice page. Hopefully this was helpful. I think I was a little tired, but good job, guys. And the labs are our number one resource. So good luck, and you guys can do it. You've taken hundreds of exams at this point, maybe thousands. Who knows? So it's just one more. All right. Bye, guys.